So welcome to the future of life science. And we have no less a grand ambition than to discuss the future of the whole of life sciences and the role that big data has to play in life science and in medicine and in healthcare. And arguably big data is an ongoing revolution which I think may be unmatched in the history of biology. So as Stefan said, my name's Adam Rutherford, and I am a geneticist, a lecturer at UCL, but I also have a public-facing role as a science communicator for the BBC, and I write books, and that's my role for the first 3 sessions is as a moderator to encourage the conversations, to introduce the speakers, to moderate the questions, and to keep you to time. That's the most important thing. We are on a super tight schedule, so I'm not going to waste much time. Stefan mentioned there are going to be 4 sessions. So the first one is going to be called From Data to Insight. So it's how we get from raw data to clinical relevance to epidemiology. The second session is going to be on how data is actually used. So that's going to include discussions about oncology and drug development and healthcare, and also that thing that we've been living through for the last 2 years, which, as I said, has fundamentally changed the nature of this meeting itself. And then tomorrow we're going to be talking about the impacts of health data for society, artificial intelligence and its role in developing data as it faces society. And then finally, I'm going to step down and Richard Coburn's going to take over, and we're going to talk about the risks and the opportunities and the ethics for people in society as the changing landscape of data management and data acquisition goes on. So in the room, we have just an incredibly wide-ranging group of people. We have loads of scientists from both the private and the public sector. We have epidemiologists, doctors, geneticists, microbiologists, and we also have philosophers and artificial intelligence experts. And in the audience, we have journalists and policymakers. And so there is an enormous range of people, experts, to discuss this very grand ambition to try and work out a conversation, an ongoing discussion about how big data and data management and data acquisition and data utility occurs now and in the future in Sweden, in Europe, and for the whole of the world, for the whole of the life sciences and biology. So that's it really from me, because we really want to get on, and there's a lot to get through. So let's crack on and introduce the first speaker, who is Mathias Uhlén, Professor of Microbiology from the Royal Institute of Technology. Mathias.
So thank you very much, Chairman. So I will try to dive into a little bit about my sort of view on data of the data revolution in life science, but I will also focus a little bit on my own work, because I still think it is nice to show a little bit what you can use for this. This is a movie that we made at the SciLifeLab. The green thing here is staining a protein called insulin. So we're actually inside the pancreas, and you can actually fly around with these new technologies and see where are the proteins and the nerves that you see in red. So what we're going to talk about during these days is the data revolution. And at least in this case, it has been defined as the 4th industrial revolution coming after computers and internet, and that is using data in different ways. I want to remind you that we have this fantastic achievement that was published last year, and that was from Google DeepMind, which is actually a UK-based effort, where they used artificial intelligence and machine learning to actually determine, or at least predict, the structures of all human proteins and also from others. And I don't have time to go through that, but it is an absolutely fantastic achievement that has taken at least 50 years, and it is a revolution for all of us working in this field. I was the founding director of the Science for Life laboratory, which is in Stockholm and Uppsala. And actually this was started in 2010. And the idea, the sort of founding principle of this was that we should use the technology which is evolving, and then we should use big data in a new sense. And this started in 2010, and now we are actually close to 1,500 researchers. So I'm very proud of this. This center has just got a fantastic funding from a non-profit organization here in Sweden, the Wallenbergs, and actually 3 billion Swedish is then devoted to data-driven life science, and I think that's very timely. The reason it's timely is that we are now on the verge, on the edge of getting a holistic view of the building blocks of human biology. This sort of started already in the 40s with the Krebs and others for the biochemistry. It was this huge effort, the Genome Project, that was sort of reported in 2001 or 2. But now we're moving into the real stuff, and that is the proteins, which are the real building blocks of human biology. It's been really hard to study these, but they are of course the targets for all pharmaceutical drugs, and we now have the tool to study them in a comprehensive manner. There is a lot of efforts now going on for mapping of all the human building blocks. And I just listed some of them here. A lot of them are from the US, and they are all quite grandiose. And they all have funding from, well, most of them from non-profit organizations, which is quite interesting. One of them is the Human Protein Atlas, which I'm leading here in Sweden. And again, this is funded from a non-profit organization, the Wallenberg Foundation. What we're trying to do in this effort is to build an international core resource to explore human biology and diseases. It is hosted at the SciLifeLab. And just to give you an idea about the effort to try to do this is that we have more than 15 million web pages that one can explore in an open access resource, and about 10 million annotated tissue images. We're very, very proud that this is one of the most visited biological databases in the world, and we are now trying to use this to explore human biology, but also disease. A lot of this effort is what we call systems biology, which is actually data-driven, artificial intelligence kind of work. I'm not going to go through this, but I think the new thing for us is that we are generating our own data, but we are at least half of the data that we're using, we get from the public, from open access. So a lot of this is to integrate your own data with data which is out there and you can download from different servers. We also have some fantastic new technologies. One is that we can study single cells in a completely new manner. And just a few months ago, we published a paper together with the Chinese researchers where we are looking not only on humans, but in this case, the primate, the macaques, using this single cell analysis. There's also a lot of new technologies where you can actually go in and look at the cells and what is happening. And I just wanted to mention here that we have a paper in Cell this week where we are looking at this with these new technologies. So why is this so important? Well, one of the reasons is, of course, to know about human biology and understand the creatures that we are. But another important part is, of course, to understand what happens when we get diseases. Can we move into what is called precision medicine, which we will talk about later here, where you actually look at in a more individualized manner. And a lot of this is about integration of data from different sources. And I don't really have time to talk about it, just to say that the objective for the future is to have the right treatment to the right patient, more targeted treatments, and have better diagnostic methods for analysis of health and disease. This we do by doing what we call multi-omics, which is actually to combine data from very many different sources and then create a study, for example, COVID-19 patients and how they can be stratified. We have been looking a lot in extreme preterm babies and see what is going on there. Also to stratify patients for drug treatment to try to say which patients should be treated with different drugs and so on. And here you can just see some of the publications from our group. What we're trying to do now is to actually move towards a pan-disease blood resource. We have now fantastic new tools that has been developed here in Sweden called Olink. And with these tools, we can actually take small blood samples from patients and then provide a detailed and comprehensive map of all the proteins and the metabolites in bloods across basically all diseases. This would be unthinkable just 3 or 4 years ago, but now we have the tools to do this. So we started this effort in a collaboration with Sahlgrenska in Göteborg, a biobank called Scapis, where we basically took 100 individuals that were healthy and followed them for 2 years, taking a lot of samples every 3 months. And I don't have time to go through this, but I think these 100 persons are the most studied persons ever on this planet, actually. And we have got a lot of information. And one of them, one of the take-home messages from this is that each one of us in this room, but also listening to this, has his own unique protein profile in the blood, sort of a fingerprint. But also this unique fingerprint protein profile is stable at least during 2 years, and we kind of expect it for the whole life. So this is, of course, very interesting. This is the observation. What does that mean for who we are, what diseases we get, and so on? We also now, very much inspired by this, going into diseases. So thanks to the fact that in Sweden there is a lot of different biobanks which are very exquisite in quality, we have then decided to select 100 diseases, 100 patients from each disease, and then do this kind of protein profile mapping in this new manner. And altogether about 10,000 samples. The technology is really expensive, so this has been not that easy to get the funding for, but thanks to these foundations, we are now going into cardiovascular disease, as you can see in blue, different ones, the cancers, the infectious diseases, neurology, Alzheimer, Parkinson, and so on, psychiatry, and also different autoimmune diseases. And we're now going into the details, and this is the sort of only data I will show. This is brand new data, a few weeks old, where we have looked at more or less all the major cancers, and we've asked the question, are they different? Can we actually see which has which cancer simply by taking 5 microliters of blood, analyze it with these new tools, and can we predict if a person has cancer or not? And if he or she has a cancer, what kind of cancer is it? And without going into the details, it is very, very encouraging. This is new data, and we, for example, for this leukemia here, for breast cancer, for glioma, brain cancer, colorectal cancer, we can distinguish the patients, the cancer patients in blue from the healthy controls. So we are sort of starting then to feel that we have a good way to find cancers. And obviously we now have to validate this in larger cohorts, but also see, can we find the cancer before we have any symptoms? And can we do it for more or less all the cancers with donating 5 microliters of blood, sort of maybe annually? So my concluding remarks then is that we have new state-of-the-art tools for plasma profiling. The fantastic thing for me, which I'm very proud of, is that these have been developed in Sweden but are now swept through around the globe in Uppsala. These tools are suitable for next generation precision medicine. We're entering a completely new era, I think. And it is possible to explore patient profiles in a sort of pan-disease manner now. So this is, of course, very encouraging. Just want to mention that I said that the funding is from non-profit, the Wallenberg Foundation, but we also have from other non-profit organizations, as you can see here. This is where we are in this beautiful city called Stockholm, at the Science for Life, which is with these other, the KTH, Stockholm University and Karolinska. And before I end, I just want to show you another movie that we have created at the Karolinska in my group, using these incredibly good new technologies to actually go into the body and look at single cells and single proteins in a sort of helicopter view. So I thought we have made 18 movies like this. I thought I would show you one on cancer.
Breast cancer impacts over 2 million women every year. It is the second largest cause of cancer-related deaths among women. But now there is new technology that gives hope for a new understanding of this deadly disease. Images in 3D make it possible to look from different angles at a tumor and see different features of the tumor, to see where and what the immune cells are attacking. The new images give enormous possibilities for future cancer research.
Thank you very much.
And if I could bring up the second speaker, which is Janne Lethiö, who's going to talk about genes. Genes is good, proteins not so much, and precision medicine. Thank you.
Thank you very much, and it's great to be here. Thanks for the invitation and a fantastic setting, and very, very important area to discuss. And this is actually the shortest time to talk about this broad topic. So I will need to discuss that during the time that we have. But just to introduce a little bit background, I'm from the SciLifeLab, scientific director there now, and my research group is on the SciLifeLab. Mathias already mentioned about SciLifeLab. That's one of the 3 big research infrastructures that we have in Sweden, where we actually are a bit different from the large physics infrastructures. We collect lots of bioscience technologies in one place, and that creates synergy to work on the environmental side and precision medicine and so on. So we can tie together lots of these technologies to understand from genome to phenotype. And since this year, we are truly national infrastructure with the sites in all the main locations. So that gives a great opportunity, not only for the Stockholm region, but also for Sweden. And I'll just show you this picture so you understand where I come from. This is my group. So we have our technology development in SciLifeLab, and we disseminate those technologies in the regional and national facilities. But I also have a laboratory in the hospital where we develop diagnostics to look at the proteome-wide diagnostics in cancer and many other diseases, and also work as a researcher at the Karolinska Institute side. So this is the intersection that my group is working. So data-driven life science, this is one of the themes of this conference. And why is this such a hot topic right now? And why do we want to work on that? And that coincides with several different things. There is an exponential increase of the life science data. But there is also huge increase in capabilities of understanding and processing and working on that data. And And these also converge with other things of sort of interdisciplinarity that is going on in many fields, and automation to increase the throughput. So we have in life science, we are turning the tables. Often we look at the data in the public domain, and based on that we form a hypothesis and design the experiments. Before we design the experiments and then maybe validate it using the data. And this is one of the reasons why we have this DDLS project, which is very exciting, and Mathias already mentioned it. It's a Wallenberg Foundation financed 12 years program together with the SciLifeLab to catalyze this data-driven life science. And you have seen already this picture. I've already added Olli here, who is chairing the DDLS project. But in the DDLS project, we have 4 areas: cell and molecular biology, precision medicine and diagnostics, that I will talk a little bit more about, evolution, biodiversity, and epidemiology and infectious biology. But the most important thing is that we will use lots of this money to recruit talent to Sweden. We will also build platforms that will connect and facilitate the team science. So this is something that will create exciting opportunities for us. And we are, of course, not alone in this, but we are quite early in this, and it's a big investment for Sweden, but there is also these type of investments and initiatives popping up around the world. And so this just shows how important the data and the data mining in life science is. So what does this mean to the patients then? And how do we convert this into a precision medicine? This is Karin that got lung cancer a few years back. And unfortunately, spread lung cancer, which is a really bad news. And only 5% of these patients survive 5 years. So what happened then is that the treatment started with intense chemotherapy. But just then, also the precision medicine is starting to roll into the clinical trials and use. And Karin happened to have ALK fusion protein that is driving the tumor, and there was these new treatments for that patient group. So that was a good situation in the sense that we could implement. And this is one of these things that happened from the initial discovery. In 2 years' time, it was used in the clinical trials world round. Not 10 years, not 15 years. And this is what is happening in the precision medicine, if we can exploit these technologies. So then Karin unfortunately got relapse, but there was next generation ALK inhibitors and new treatments with precision radiation therapy. And this is the situation today. I met Karin just 2 months ago, and she told me that her quality of life is good. She's enjoying the life, but she's also telling me, we cannot wait. This needs to be implemented broader and faster. So this is something that we are all here to catalyze as well. And lung cancer that Karin had is a very, very good example of what is actually happening here. This is an evolution how we view lung cancer. So 1990, lung cancer was viewed as one disease. And then with the immunohistochemistry and histopathological analysis, it divided into subclasses. And then we are slowly entering into the molecular medicine area, where we look at the driving mutations and genomics. And that's first time we can also couple precision drugs into the, just like I told with the Karin's case. And now we are heading into coupling much more data into that. And this is needed. This is the new drugs and immunotherapies in lung cancer. And what we know is that the single drug is not enough. We need to be able to combine that. And this is a game changer, because one mutation and one drug is not going to solve this. We need to start coupling other data types in order to understand what combinations do we work on. So This is something that we and everybody else is starting to work on, piling out this data together and systematize this analysis in relation to the patient outcome. And this is something that we have been also working. So this is a slide from our group where we actually did epigenetics, genomics, transcriptomics, proteomics, and histochemical analysis of the tumors in one go. And what we can see is that deep molecular phenotype, the proteome, actually clusters these patients into particular groups. And when you start looking at these groups, the genome alterations are not randomly here. The histopathology is not randomly distributed here. And by mining this data, we can start building these treatable combinations here. The other thing that we can do is that we can also look at the novel proteins that the genome aberrations in cancer create and use that for cancer vaccine development. And that's been done before by predicting that from the genomics. And this is the first time we can do it by actually looking the antigens. And this is something that we have been testing with our collaborators in Helsinki. And they have developed the vaccines and are working with the mouse models that are very aggressive. And when they got these vaccine candidates and developed these vaccines, they told us they've never seen such a response controlling the tumors. So it just opens up new possibilities by coupling these layers of data together. The other thing is, of course, that it's only part of the process in the healthcare when you get the tumor diseases. You also need to find the patients and monitor and manage the treatment and relapse. So one of these things that we have been working on is to turn these multi-layer analysis into a screening tool. And what the problem is that we have great ways to find lung cancer in large-scale screening processes with the CT scans. But it causes lots of radiation, it's quite expensive, and it has horrible false discovery rates. So we do that every 2 years, but that doesn't really reduce the mortality because it's too seldom. So what we have done is that we have teamed up with the nursing researchers that did deep interviews of 60 lung cancer patients and noticed that this disease is not silent, but the symptoms are quite generic. So based on that, we developed a questionnaire that we tested on 1,000 patients, so 1,000 cases referred to lung cancer elucidation. It's very easy in a smartphone. That gives us quite good prediction which population of patients can be potentially having a lung cancer. Same time, we took plasma samples of these cases and developed proteome assays. That's also quite good to narrow down. So this is a way to filter patients into the screening studies. Instead of screening everybody, we can maybe find the patients. And this is also a use of then funneling, and then next step, try to use the precision medicine to decide the drugs. And the other thing that we also noticed when we started to work in the precision medicine in the clinical trials is how do we interpret this data to the clinicians, to the group of people that actually make the decisions? So that's out of the necessity when we started to work in a large European genomics-driven trial, we generated a molecular tumor board portal. So there is a big genomics trials in Europe that does cancer sequencing of patients. All that data is then sent to SciLifeLab, and we analyze, harmonize that data, and generate patient-specific report. That report is then discussed in the molecular tumor board once a week across Europe, and patients are referred to the clinical trials. That way, there's a couple of things that have been really eye-opening for us. One thing is that gathering around this report, we can actually form a community across Europe where that exchanges information patient-centrically. The other thing is that it's been really great tool to catalyze the the precision medicine process and the team science, where the bioinformaticians and the clinicians and researchers are developing this together. And our next sort of roadmap is to not only do the single genomics markers here, but add proteomics data, add immunohistochemical data, add pathology data, and so on into this tool to move to the combination treatments, to move to a systems medicine, and same time sort of introduce new technologies, new data types into this. And I think that it was already mentioned that the pandemic has been really changing how we work. And this is an opportunity for precision medicine. What has been happening is that we share much more data. We have a sense of urgency of finding solutions. There is new teams, new sort of data portals and ways to collect the data together. And this is something that we just need to convert into the precision medicine. And in Stockholm region, we have formed Precision Medicine Center Karolinska that tries to break these walls, brings in technologies, brings in clinical trials, brings in routine changes, clinicians, radiologists, researchers, and so on. And this is something that has been very good learning experience to understand what needs to be done. And there is a lot to be done, but there is also lots of enthusiasm. And also from the SciLifeLab side, we have formed precision medicine team that is trying to connect the technologies that we have in more really relevant setting in the clinical trials, and also the whole data-driven life science precision medicine area, which I'm heading into this, so that we can connect. SciLifeLab can dock into the university hospitals and healthcare. So we will try to catalyze this. So with that, I'll end up my talk that we usually say that the precision medicine is not revolution, it's an evolution. We have been aiming onto it all the time. But all these new technologies, all the data that we have, all the capacities that we have, it's actually forming completely new opportunities, and we are entering into a new field. So with that, I thank you for your attention. I'm happy to discuss with everybody.
And welcome to the stage, Cecilia Magnusson.
Ska vi se. Yes, thank you so much for having me. I will now take a completely different perspective on health, namely that of health from society to cell, as opposed to from cell to society, as we might have spoken of hitherto. So this is me, Cecilia Magnusson. My day job is as a director of the Center for Epidemiology and Community Medicine at the region of Stockholm. So I'm responsible of serving our system with epidemiological data of how the population of Stockholm is doing, and what we could do about what's not going so well. I'm also working as a professor in epidemiology at Karolinska Institutet. And we all have these very unambitious titles, don't we? So mine is What Determines Our Health? But I hope I will be able to answer this question. So when we ponder about the determinants of health, it might be useful just to have the backdrop of what are the disorders and diseases that cause our ill health. So this is data from the Global Burden of Disease project, depicting now the contributors to something called DALIs. And DALIs is a composite measure accounting for premature mortality as well as disability. And as you can see, and we all sort of know, I think, that cardiovascular disease and cancers, they do contribute to a lot of DALIs. But what we talk perhaps less of is, I think, musculoskeletal disorders. It's quite high up on the agenda, and our treatments and our understanding of the pain disorders is still quite poor. Well, having shown you that circle diagram, I must then add it's all wrong, because then came COVID. And we added now this is the number of deaths in Sweden by cause of death in 2020. And I just wanted to remind you that COVID added substantially to the other causes of death. And we know now that the last majority of that number, the orange number there, were actually excess deaths in Sweden. So as if anyone didn't remember about COVID, that was that. Okay, so we're talking a lot about precision medicine. It's extremely interesting and it's so promising, it's so impressive, the previous speakers here today, of what will come with the future. But I just wanted to remind you about the saga of the decline of the coronary heart disease mortality. It has actually declined by more than 70% since the 1980s. That's a lot. And how did that come about? Well, it's not an easy task to evaluate that, but this is one paper. There are others that come out in that ballpark type of data, that prevention did more of this job than did the clinical approach, sort of treatment of existing disease. So prevention there would be the falling smoking rates as the number one. But there are other very big contributors, as in, for example, a bettering of our blood lipid levels, for example, and also breakthroughs, as we have forgotten, namely, for example, treating high blood pressure is very important, and also treating pre-diabetes. Okay, so another sort of backdrop. If it's not then mostly medical care, but more prevention and population level individual risk factors, what are those? This is data now again from the Global Burden of Disease project, listing the DALIs, how they are generated by different individual risk factors in Sweden in 2019. And I wanted to draw your attention to, while smoking rates have fallen such, it still has its top position there among the determinants of DALIs. Then comes high blood pressure, dietary risks, high fasting plasma glucose, high body mass index, and a little bit further down there on the list you find low physical activity. And sometimes I think we talk lots and lots about physical activity and how fantastic it is. And it is fantastic, and it generates lots of health. But I think in Sweden, maybe Anders Hansen have been a little too effective as a communicator. So now physical activity is sort of the answer in Sweden to every question, and that may not be necessarily true. Okay, so it seems now if we then get an excellent healthcare system with precision medicine, treating all the new lung cancers, and a super healthy lifestyle, doing lots of physical activity, will we be fine then? Will the health on the population level be fine? It will probably be pretty fine. But my point is that there are other things that matter too. This is a bit of a complicated, it's data from my own research and hence a bit complicated, but it's the risk of autism autism in the child in relation to whether the child was born preterm or postterm, and whether it was born small for gestational age or large for gestational age. And it's actually, there are very high risks of severe autism in children who have disadvantaged obstetric circumstances. This is just to draw your attention to reproductive health, maternal health as a basis, as a foundation for public health work. School matters too. School matters for public health a lot. This is data on suicide attempts and school results. So young people and suicide attempts, that's a big problem in Sweden. We have high rates of mental ill health in young people, high rates of suicide attempts, and we know that this is disabling and means a poor prognosis in terms of health and social outcomes. So these suicide attempts is an interesting outcome. And we think that we know that school results is an important predictor of suicides and suicide attempts and mental ill health. So that is the graph to the right. The GPAs, the compulsory school leaving grades, how well you do in årskurs 9. And that you see that the worse you do, the poorer you do in terms of suicide risk, suicide attempt risk with, again, very strong relative risk, very high risks there. Then one would think, of course, yeah, that's because the people who doesn't do well in school, they have so low cognitive ability. So in the other graph, it's the same data, but then the compulsory school leaving grades are adjusted for IQ at age 13. And that just doesn't change things much. So this is some type of evidence how success in schooling, a good schooling in itself, is very important. And it goes on. So it's not maternal health, schooling, and your own income matters. This is data from our center. It's not been released yet because it's new data on COVID. It's the incidence and relative risk of hospitalization for COVID-19. And in the left-hand graph, you have the incidence according to the different quantiles of income. And the yellow one in that graph is the people with the lowest level of income, and the number of hospitalization then throughout the waves of the pandemic in Sweden as it panned out in Stockholm. As you can see, these curves are very different. They're quite shifted and in quite a gradient there with the income level. In the right-hand graph, it's the same data, but it's modeled as relative risks. And now it's also adjusted for very important factors such as your sex, your age, your country of birth, your area of living, which was important because the virus clustered geographically. Your occupation, that was also important, and your vaccination status. And regardless of all of that, you still have an increasing social gradient in COVID-19. Oh, so now something different. I just want to read this to you. Say this city has 10 million souls. Some are living in mansions, some are living in holes. Yet there is no place for us, my dear. Yet there is no place for us. Once we had a country and we thought it fair. Look in the atlas and you'll find it there. We cannot go there now, my dear. We cannot go there now. That was W.H. Auden and his refugee blues. And it goes on. This is just the two first verses. And why do I read that one? Because for me it's the sentiment about migration and some backdrop to us why migration is such an important determinant of health as well. And we live in a new era of migration, and we have the war in Ukraine, so migration is certainly something we have to think about when we think about health and public health. Again, complex data from my own work. It's again about autism and severe autism. The red line is the relative risk of severe autism in the child in relation to your mother's migration, the timing of your mother's migration in relation to pregnancy. So around the note here, this is around when the mother had migrated around the time of pregnancy, and that is where the peak of autism is, the autism risk is. So there's something pregnancy-related, that's something adverse with migration that might actually affect the child in the womb, we think. On the individual level, social factors then like schooling, like your own income would matter a lot. But not only on the individual level, but indeed on the societal level. So the level of welfare in the country is tightly, of course, connected to the level of population health in that country. This is, I think, was striking data from England, and suicide risks associated with the economic recession in 2008 through 10. And it's you can see here the suicide rates. The blue is the curve of the unemployment rate over time when the crisis happened in men and in women. And then the red line is the suicide. And this is what we know from suicide epidemiology, that we have done a great job in lowering suicide. But that saga came to an end here, and suddenly we had a rise again in suicides in men. Not in women that much. That's why I put the and gender in brackets in the title of this picture here. But I think it's amazing that a financial crisis can have direct effects like this. And the mad cow disease, you remember, there were more suicides than there were people who was killed by the actual pathogen. So the societal level matters. This determines our health. So that's the answer to the question I was supposed to give. It's the onion. Our hosts have named this model the onion, which I think it's really good. It's more formally referred to as the rainbow model, or the Dahlgren and Whitehead model of the determinants of health. And all of these levels in the onions, they determine our health, and they interact with one another. And then I put a little thingy on the side there with the outermost layer of the onion, the general socioeconomic, cultural, and environmental conditions, and there we can, well, think more and more about that little thing there, the climate change that will have, of course, the biggest impacts of all. Thank you.